So maybe quick wrap up on the orthology session. Any um, questions or comments?
Let me respond to a question from Zoltan in the about functional annotation. It sounds like you have some interesting things to do in for functional annotation based on eggnog. Eggnog is a useful orthology detection method, particularly for looking at domains and therefore useful for meta-transcriptomes. My suspicion is that the new release of OrthoMCL would be more effective for meta-transcriptomes, but we haven't tested that out. So I'd suggest, Zoltan, if you're interested, to contact us directly. You're welcome to email Stuart or myself, but even better would be to click on the Contact Us link so we make sure it gets routed to the right place.
I have done a couple demo runs with meta-transcriptomes, and it works really beautifully.
And there's some interesting thoughts about how that would work compared with eggnog, which is not really an orthology detection method. It's a domain-based orthology detection method rather than a protein level orthology detection method.
It's broader. Their groups are bigger. Ours are more specific.
Right, but that is because many proteins have been assembled from multiple domains that may have different sources.
One question that came up in our group right in the end from Bibo was that this one group she was interested in OrthoMCL6, and she tried to find it in OrthoMCL7 with the group ID, and she couldn't find it. That was curious because we were able to find it with find a group in Ortho7 with the gene ID, and it seems to me that the Ortho6 group ID should have mapped, and so that's so I have
yeah. That's either a bug or some little thing like you've added a space when you pasted it in, and our thing is being
right. So we need to follow up on that and double check on that. Yep.
Yeah, I mean it's worth double checking. I've looked up a lot of Ortho6 IDs, and it has been working.
It's not working with several that I tried. In my group, we encountered the same thing. I entered a contact us link there just for people on the call. We use contact us as well as a way of entering things into a tracking system. So the ID was shown on the OrthoMCL7 page, but
it's
did
the
not
site
work.
search that's not working, or the look up the ID that's not.
I think it probably wasn't fully. My guess is that there's a bug with the site search indexing, which would be good because that would be very easy to fix.
Yeah, because there's a separate lookup ID search, and you could try it there as well. But I will look into this, obviously.
It's unfortunate that that's one that the exercise was built on, but it's on the other hand, maybe it's fortunate because it highlighted it. This is why this is a beta release.
Right, it's a beta. We're still working out the kinks.
So we are happy to have any comments from you about things that you do or do not like, or ways you would like to see the phylogenetic tree views, or other things that you might like or not like or miss from the previous version or would like to see in the context of other ortholog searches.
So could it be because in the 7 beta version, actually the ortholog group number is changed?
The ortholog group number is changed, but if you scroll down the list, there is a place where previous ortholog groups are listed. And in this case, in the case that we used, the case that was part of the exercise, that number is there. So it's clearly just not being indexed appropriately.
I see. Okay.
But yes, you're correct. The ortholog group number has changed. Ortholog group 7 number OG7 1 2 3 4 5 has no relationship to OG6 1 2 3 4 5. And you might think that that's unfortunate. You might think it would be nice to maintain that numbering, but as you'll imagine, well, we could potentially do so for new versions of genome annotation where there's usually a one-to-one mapping. Ortho group in new implementations with new species, groups are broken up and merged together all the time, so there are virtually no groups that are 100% identical between one release and the next. That they will be largely identical. Probably in this particular case, I took a look at them and it looked like at least 90% of the proteins in the old ortholog group are included in the new one and vice versa, but not 100%.
One thing I wanted to highlight quickly for everyone is that with these trees that are pre-computed, you can actually download the Newick file format. And that is quite nice because you can then, if you take a look at that format, obviously it's you know, most people will not read this, but it contains all the information to build a tree, and you can put it into many different softwares and tools that you have. And so, for example, I took this tree right here, downloaded a Newick file. I just copied and pasted the sequence into ITOL, which is a free resource, and I was able to, you know, very quickly just draw a circular map of my tree. So for some of your trees, this may be very nice to do, and you can then play around with it. And ITOL is very interactive and functional, and it may. I sometimes have these open side by side because I personally have a hard time visualizing a big long tree when it's sort of going down the list, and so I find it useful to actually then take the tree into another program and play around with it there.
Sorry. But the page, sorry, I don't speak English very well, but in the exercise, at the end of this strange map that we have a link to, I don't remember, we have to make a tree, and it takes you to the iTool, to that page that there's no need of, it's a rank cluster omega for selected. So you have to select what you can select all at the top, and then you click run cluster omega, and it will take you to the iTool that you should show.
Yeah.
Yes,
okay.
that's correct. You can.
So you don't need
get,
to download the.
That's true.
Oh, right here.
you,
That's right.
yeah,
to
yeah,
click here to view. You're right.
I reached to the end of the homework.
That is good job. Good job.
That is a perfectly valid way to work.
Yes.
It's different than the tree that we make with every organism in the ortho group, with every protein. So it's not a subtree, it's a newly calculated tree. So they can be different. It's not wrong or right, it's just a little bit different.
So if you did what you suggested, Maria, it should be exactly the same, because Maria suggested selecting all of them.
Yeah,
and that would yield the same result. Although technically, what you are doing is unnecessarily duplicating computation, because we've already generated that tree and provide you with the Newick file, whereas you have recalculated the alignment. Fortunately, Clustal is pretty fast, and this is a relatively small tree, so that's the wasted computation is not great. So what you've done is exactly the same result that what Omar got. Actually, but as Stuart indicated,
it's a tiny bit different because we align with MAFT instead of cluster.
That's correct.
And we build a maximum likelihood tree, not a maximum likelihood, rather than a cluster tree.
Yeah, good point.
So
good
they're slightly different.
Okay. But it takes you to, I mean, you run the cluster and then you can see it instead of, well, you can see it in different forms, in a graph
neighbor
form.
joining tree. So it's a little different. Again, not right or wrong, but
that is a good point. They should be very similar, but possibly not identical, and it runs an additional computational step. There's the big advantage as well, though, of doing it the way Omar was doing so, is that if you don't want to see a tree for all of these taxa, and you very likely do not want to see a tree for all of these taxa, you may not want to see a tree, for example, that includes 20 different strains of Plasmodium falciparum, that is only restricted to the reference strain. You can select what you want and generate that tree. In any case, the ITOL tool is a very useful tool, as Omar indicated. As it happens, it was generated by the same group that was responsible, or the same lab that was responsible for the eggnog domain ortholog predictions that Zoltan Fuesi was commenting on previously.
Stuart, is the methods that you described for the way we generate a tree, is that are described somewhere on our, because we're
Yeah,
again, as a beta side, we're missing some things or we need to add more information, but yeah.
Yeah, but it is there, but it could be made more obvious.
Okay, cool. Yeah, we should do that, certainly.
Vasilios, there's a
Yes, thank you for the presentation and for the information. I have a question. When you query OrthoMCL and you get a list of orthologous groups, orthogroups, is it possible to download all sequences within the groups in one go, or you
Yes.
have to go and do it one by one?
There's a download button at the top of the group page.
So
and
let
you
me-
can figure
let me
that
try
to download
Okay.
sequences. You can also convert the ortholog groups to sequences and download those.
Yep.
Okay, so is I cannot see, okay, so the description is the description of the sequence for the output option, or the group, the orthologous group description?
It's at the very top of the group page. There's a download button.
No,
Yeah, I mean, when you have the options. Okay, I did it, so yes, I got the information I want. Thank you very much. That was
you
helpful.
found it? Okay, all right, great.
Great.
I was just about to show it. Okay, perfect.